Welcome back to this next video in which we are discussing inflammatory bowel disease. Okay, so in the previous video we studied the structure of the wall of the small intestine, and remember at the end of the previous video we said that to adapt the picture to the large intestine, all you'd have to do is take away the villi. So imagine taking away the villus on this picture and just having a flat epithelium here, still with the crypts of Lieberkühn, and that would then be a picture of the wall of the large intestine. So in fact, the epithelium of the large intestine is actually simpler than that of the small intestine, because rather than having evaginations and invaginations, we just have the invaginations now. So the epithelium is still a simple cuboidal epithelium. We call the major cell colonic epithelial cells rather than enterocytes, but it's the same sort of cell that you're going to have still with microvilli, and you'll still have goblet cells interdispersed amongst the colonic epithelial cells. In addition, the epithelium is still being continuously turned over, so at the base of the crypts of Lieberkühn, you're still going to have intestinal stem cells that are dividing to produce cells that will become the colonic epithelial cells and the goblet cells. Underneath the epithelium, you still have a lamina propria with the blood vessels and the lacteals in the large intestinal wall. You still have the muscularis mucosa, you still have the submucosa, you still have the muscularis propria, and you still have the serosa. So everything is pretty much exactly the same, except that you don't have villi. So, we've now studied the structure of the wall of the small intestine and the large intestine, which sets us up beautifully to discuss the pathophysiology of inflammatory bowel disease, because those are the two major portions of the gastrointestinal tract that are actually affected by inflammatory bowel disease. So, let's now move on then and discuss pathophysiology. So we'll come down to a nice, clean portion of the board. So before we move on to pathophysiology, allow me to give you a little speech here that is important. And this speech is about the fact that science is just models. Science is models to help us understand the reality in which we live. I am going to present you a model of the pathophysiology of inflammatory bowel disease. It is a theory that helps us understand inflammatory bowel disease, and which we hope bears some similarity to what actually does happen. However, be aware that the story that I'm going to tell you does have holes in it, and it may well change within the future. We still do not have a full understanding of what happens in inflammatory bowel disease, and I can only present you our best current model. So the two warnings are, this story is going to have holes in it. There are bits that I'm just going to have to say, you're just going to have to accept that that is what happens, and that what I'm saying may well have changed in 100 years' time. If I'm still alive in 100 years' time, I can hope. Okay, so let's begin then. So the first thing to do is to kill a major myth surrounding the pathophysiology of inflammatory bowel disease, which is that it is not anymore believed to be an autoimmune disease. So I'll say that again. Crohn's disease and ulcerative colitis are no longer believed to be autoimmune diseases. We used to think that they were autoimmune diseases and that the immune response was attacking part of the actual intestinal tissue, i.e. that it was a self-attack, it was an autoimmune attack. However, we no longer think that. But it remains a common misconception that people have to think that Crohn's disease is an autoimmune attack on the intestine and that ulcerative colitis is an autoimmune attack on the intestine. We no longer think that. Our best current model is that they are no longer autoimmune diseases. Okay, so there's the first thing to say. So if they're not autoimmune diseases, what do we now think they are? The answer is, we think that people with inflammatory bowel disease are attacking the commensal microorganisms within the lumen of the intestine. So, I mentioned the commensals earlier on. Now let's discuss them in a little bit more detail. So, starting by writing down the word. So, inside your intestines, the small intestine and the large intestine, there is absolutely loads of microorganisms within the food that is moving through the intestine. And these microorganisms are not dangerous. They do not cause you a problem. Instead, they actually help. They help with digestion because they digest things that are in the lumen of the intestines and actually help us break things down, and they help us absorb some of the nutrients. Of course, they're taking things as well, but they help us break down things that otherwise our intestine would fail to break down. So they're actually very useful, these commensals. So when you have a microorganism that is not dangerous and is actually helping the body's function, we call these commensals. So the commensals that are within the intestinal lumen would be known as the gastrointestinal commensals, or the intestinal commensals, the commensals within the gastrointestinal tract. Okay, so what microorganisms are actually present within there? Well, you have absolutely huge number of different species of microorganisms. Too many for me to list. You know, you can go on the internet if you like and look these up. You will get lists of hundreds and hundreds of different species of microorganisms that are present within the lumen of the intestine and which help to break down our food and which are considered commensal microorganisms. Let me just give you some of the broad categories, however, and I think I'll change colour. So, they're mainly single-celled organisms, and there are two major different categories of single-celled organisms that we're going to have. The first are the prokaryotes, and these are single-celled organisms where the cell does not have a nucleus. The DNA is instead just in the cytoplasm. So remember, a prokaryotic cell is a cell that doesn't have a nucleus. And there are two major different types of prokaryotes. Bacteria, of course, which are the famous types of prokaryotes, but also a slightly less famous type of prokaryote, which are the archaea. So, bacteria and archaea, then, are both prokaryotic categories of living organisms. They are single-celled organisms that do not have a nucleus. And within the intestinal lumen, you are going to have a huge number of different species of bacteria and a huge number of different species of archaea. So these are huge categories with loads of different species of microorganisms that are all prokaryotes. Okay, in addition, you're going to have some eukaryotic microorganisms, and I've spelt this wrong already. Let me just reverse this. Eukaryotic, single-celled organisms as well. And of course, eukaryotic cells are those that do have a nucleus. Our cells are eukaryotic cells. However, of course, we are not a single-celled organism. Examples of single-celled eukaryotic organisms are protozoa. So these are huge categories of living organisms: bacteria, archaea, and protozoa. They're all single-celled organisms, but bacteria and archaea are prokaryotic single-celled organisms, and protozoa are eukaryotic single-celled organisms. They do have a nucleus. So again, I'll stress that these are massive, great categories, all containing a huge number of different species. I'm not going to go into the different species that are actually present within the intestinal lumen, because we'd be here all day, but there are loads of different single-celled organisms within the lumen of the intestine which are not dangerous, which are living there in peaceful cooperation with our bodies. They will not invade into our own tissue. They will stay in the lumen, and they are actually helping us hugely by digesting things that we would fail to digest. Okay, so these are known as the commensal microorganisms within the intestine. So, because these things are not dangerous, you should not initiate an immune attack against them. Your body should not attack these organisms. And in most people, we do not attack these organisms. If you did attack these organisms, you would get inflammatory bowel disease. So we believe that Crohn's disease and ulcerative colitis are caused by an individual attacking the commensal microorganisms within their intestine. And we believe that one of the major differences between Crohn's disease and ulcerative colitis is that you're attacking different microorganisms. And it may well be the case that in different people with Crohn's disease, they're attacking different commensal microorganisms. It's just that the type of immune response they initiate is the same, and therefore the disease presents roughly the same. But we certainly think that in Crohn's disease versus ulcerative colitis, you are attacking a different commensal microorganism. But it may well be the case that in different people who both have Crohn's disease, that they are actually attacking different commensal microorganisms. It's just that the distribution of those microorganisms happens to be roughly the same, and the immune response that they initiate is the same, and therefore the disease looks to us as though it's the same, but it's actually attacking a different commensal microorganism. So, to summarise what I've told you so far, we do not believe that inflammatory bowel disease is an autoimmune disease. We don't believe the immune response is attacking your own tissue. Instead, we think that it is a failure of tolerance, and this is a fantastic little term here that immunologists will be very impressed if you use. So I'll put it here. We believe that it is a failure of immune tolerance to the commensal microorganisms. So in most people, you tolerate the commensal microorganisms. The immune response does not attack the commensal microorganisms. We think in people with inflammatory bowel disease, it's a failure of tolerance to the commensal microorganisms. And in the different types of inflammatory bowel disease, we think you're attacking different commensal microorganisms, hence why the distribution is different and why the type of immune response that you get is going to be different between the two. Okay, so that's the big picture of what we think at present is actually happening in inflammatory bowel disease. Let's now go into more detail of how this is actually going to occur. So, the first thing to say is that in inflammatory bowel disease, we think that the adaptive immune response occurs first. We think that you get an adaptive immune response attack against the commensals, and that this brings in the innate immune response. It forces the innate immune response to attack the commensals as well. So let me just explain this in a little bit more detail. So there are two major parts of the immune system. There is the innate immune system, and then there is the adaptive immune system. Now, the difference between these is that the innate immune response is not specific. It is the response that you initiate in any tissue within the body when there is an infection present there. No matter what the pathogen that is causing the infection is, you initiate the exact same response. So it's not specific at all to the pathogen. And this is the inflammatory response. So the innate immune response is the inflammatory response. It occurs exactly the same no matter what pathogen you are actually dealing with. It is not specific at all to the pathogen that you are dealing with. It is effective in many cases. It's very effective at least clearing some of the infection. But of course, it's not as effective as the adaptive immune response, which is specifically designed against the pathogen that is actually infecting you then and there. The advantage to the innate or non-specific immune response is that it's far quicker than the adaptive immune response. So the innate immune response or the inflammatory response or the non-specific immune response, this is the initial immune response. It's very quick and it's the same for whatever pathogen is causing the problem. The adaptive immune response is much slower. This takes a few days to actually start up. However, it's incredibly powerful. It has powers that the innate immune response can only dream of having. It's very, very powerful, and it is a specific immune response, i.e. to different pathogens, you initiate different adaptive immune responses. So in response to the specific pathogen that you are being infected with, you will get a specific adaptive immune response initiated, and this is very, very effective at clearing the infection completely. So, normally what happens when you get infected by a pathogen is this one happens first. The inflammatory response occurs first, it's very quick, and then the adaptive immune response occurs after. So normally it goes in this order. You start with an innate immune response and then you get the adaptive immune response. in inflammatory bowel disease, and indeed in many immune conditions such as the autoimmune diseases or diseases where you are getting a failure of tolerance to some entity which is causing a problem. It is not this way round. Instead, what happens first is the adaptive immune response occurs first. And the adaptive immune response has incredible power. When the adaptive immune response happens, it has the power to then say, we need to initiate an innate immune response as well. So in inflammatory bowel disease, it's this one that we think happens first. This happens number 1, and then this one happens number 2. So when the adaptive immune response attacks the commensals, it then calls the innate immune response, the inflammatory response, to attack the commensals as well. So this is how you're actually going to get inflammation occurring within the wall of the bowel. You're firstly going to get the adaptive immune response occurring, and it's then going to call for the inflammation to occur within the wall of the bowel. And I'll stress again that that's not normally the way it occurs. Usually it's the innate immune response first and then the adaptive immune response. But in this case, the innate immune response doesn't attack the commensals. It's the adaptive immune response that goes wrong, and then it tells the innate immune response to attack the commensals. Okay, so let's now discuss the adaptive immune response in a little bit more detail and discuss the beginning of the story of how an individual is actually going to develop inflammatory bowel disease. So, the adaptive immune response, it consists, of course, of two major types of cells. Two major types of white blood cells are the controllers of the adaptive immune response, and these are the B cells and the T cells, also called B lymphocytes and T lymphocytes. Very, very powerful cells of the immune system, the cells of the adaptive immune system. And in your body, you have billions of these cells. They are present within the lymph nodes, they are present within the spleen, and they are present also circulating around the bloodstream. So you have absolutely loads of these types of cells. Now, B cells are incredibly powerful, however they are not believed to be that important in inflammatory bowel disease. It is the T cells that we are going to be extremely interested in. So in fact, I'm not going to talk really any more about B cells. We are going to talk about T cells because these are the ones that are believed to be extremely important in inflammatory bowel disease. So, some information about T cells. We can divide the T cell population into two major groups. So, if you have a T cell, you can label it as either one of these two. So, there are the CD4 Positive T cells, also known as the helper T cells. So we put a T and then a subscript capital H for helper. And then there are the CD8 positive T cells, which are also known as the cytotoxic T cells. So we put T/C. I will probably just refer to these as CD4 positive T cells and CD8 positive T cells, but be aware that there are these other names. So let me just say what these little subscripts stand for. So the H here stands for helper, and the little C here stands for cytotoxic. Okay, so not too complicated so far. If we look at all the T cells within your body, we can split them into these two great categories, CD4 positive T cells and CD8 positive T cells. And these two populations of cells are involved in completely distinct parts of the adaptive immune response. So there are two huge arms of the adaptive immune response, which are the one that involves CD4 positive T cells and which then commonly involves B cells. So these two are partners in crime, hence why these ones are called helper T cells, because they help B cells. So one of the great arms of the adaptive immune response is the CD4 positive T cells, and then usually the B cells, although as we'll see, the B cells aren't believed to be that important in inflammatory bowel disease. And then the other great arm of the adaptive immune response is the CD8 positive T cells. So, what are these two arms of the adaptive immune response aimed at doing? Well, let's start with the CD4 positive side. This is the type of adaptive immune response that you use when you are dealing with an extracellular pathogen. So when the infectious entity, when the pathogen is present within the extracellular fluid, such as most bacteria, most fungal cells, when they're causing an infection within some tissue, they're not usually inside your own cells. Instead, they're floating around amongst your cells in the extracellular fluid. That's what I mean when I say an extracellular pathogen. A pathogen that is not actually going inside your cells. It's inside your tissue, but it's floating around amongst your cells rather than actually infecting your individual cells. So when you're dealing with an extracellular pathogen and you want to launch an adaptive immune response against that, it's usually the CD4 positive arm with the B cells that you're going to use, or with or without the B cells. As we'll see, CD4 positive T cells, they can do things without the B cells as well. One of their great functions is helping B cells, hence why they have the name helper T cells, but they can do other things as well, and it's those other things that are going to be important in the pathophysiology of inflammatory bowel disease rather than them helping B cells. Okay, so extracellular pathogens, we use CD4 positive T cells and the B cell response potentially as well. The CD8 positive arm of the adaptive immune response, on the other hand, is the intracellular response. This is when you have a pathogen that is actually inside your cells. So this is, in a way, a scarier pathogen because not only is it infecting your body as a multicellular organism, it's actually infecting your individual cells. It's going inside your cells and infecting your individual cells. So examples, of course, are viruses. Viruses go inside your cells and use them to make loads more copies of themselves. Horrible, horrible things, viruses. Some bacteria can go inside cells as well. So it's not just viruses. Even some protozoa can go inside cells. Malaria, of course, famously goes inside red blood cells and liver cells. And it's an example of a protozoan parasite. So when you're dealing with an intracellular pathogen, obviously it's necessary to have a very different type of immune response to that, and it's the CD8 positive T cell response that you initiate there, and these cells are very powerful. The reason they're called cytotoxic T cells is they go around and they kill cells that are infected with the intracellular pathogen, and of course that seems quite drastic. But the idea is if we kill the cells with the pathogen inside, then we can hopefully annihilate the pathogen completely. So yes, a few cells will die, but it's worth it to save the rest of the cells. So we sacrifice a few cells to save the rest. Okay, so what have I told you so far? So we have two major populations of T cells, the CD4 positive T cells and the CD8 positive T cells. CD4 positive T cells are going to be initiated in extracellular pathogen immune responses. CD8 positive T cells are going to be initiated in intracellular pathogen immune responses when you're attacking an intracellular pathogen. So which population is important in inflammatory bowel disease? It's this one. I don't give a damn about these ones as far as the discussion of inflammatory bowel disease is concerned. I just care about these CD4 positive T cells. These are the ones that are going to be extremely important in inflammatory bowel disease. And you might say, well, don't they work with the B cells? But as I've already been trying to stress, they can do things on their own without using the B cell response. And in inflammatory bowel disease, it's thought that that's actually more important. We don't think that B cells are important in inflammatory bowel disease. Okay, so more of a discussion then of CD4 positive T cells. So you have a massive great population of CD4 positive T cells, and they'll be present within lymph nodes, they'll be present within the spleen, and they'll be circulating within the bloodstream. And also in other places. For instance, as we progress with our discussion of the pathophysiology, we'll see that some of them are going to be present in lymphoid aggregates within the lamina propria of the small intestine and the large intestine. So you have a massive great population of CD4 positive T cells. Let me just draw a CD4 positive T cell over here. So they're very boring looking cells. They are a nice blobby cell with a very large nucleus, which I'm drawing here. So this is representing a CD4 positive T cell. So, as I say, you'll have billions of CD4 positive T cells in your body, and here comes a very complicated but extremely important concept to understand. Those CD4 positive T cells, they are all directed against different pathogens, okay? They're all different. You have a huge variety of CD4 positive T cells. So if you were to look through your massive great population of CD4 positive T cells, so if you were to put on this board your entire collection of CD4 positive T cells in your body, they would all be directed against different pathogens. Now, the way that a CD4 positive T cell is directed against a pathogen is that it is actually directed against a specific peptide fragment. So every single CD4 positive T cell, it has a peptide fragment that it is directed against. So they are all programmed to attack a certain peptide fragment that they recognize. So this is a sequence, a small sequence of amino acids, and the idea here is that that sequence of amino acids is never ever present in any of your own proteins. If it ends up present in any of your own proteins, then you can potentially develop an autoimmune disease. So we only have CD4 positive T cells that are directed against peptide fragments that are not present in any of our own proteins. At least that's the idea. It does go wrong, of course. So all the different CD4 positive T cells, they have a peptide fragment, a sequence of amino acids, and just to absolutely get this across, let me just draw a little peptide fragment here. So this is a little peptide fragment, a sequence of amino acids, a polymer, or an oligomer, I should say, of amino acids joined together that each CD4 positive T cell is directed against, and each one has a different little peptide fragment that it's directed against. And the idea then is that these peptide fragments that your CD4 positive T cells are directed against are not present in any of your own proteins, but might be present in the proteins of a pathogen. And we call the proteins of a pathogen that you can initiate an adaptive immune response against, we call those antigens. And we write it AG, like so for short. So the idea is that you will have CD4 positive T cells that are directed against peptide fragments present within the antigens of pathogens. And these CD4 positive T cells you can then use to fight that pathogen. So let's say you've got pathogen A, let's just call it pathogen A, and I'll just draw pathogen A here just to get my point across. So let's say this is going to represent pathogen A, and pathogen A has an antigen here. So let's say that blob on the surface of pathogen A is an antigen, and I've just made this up. There is no pathogen A. This is like maths. I've called it pathogen A because it's just some pathogen. Okay, so here is an antigen. If I want to initiate a response to this, I need to look at all of its antigens, all of the peptide fragments that I could get from its antigens, and then I could use the CD4 positive T cells that are directed against peptide fragments of the antigens of this pathogen to attack this pathogen. Okay, so that's the basic idea. Very complicated concept, very important to understand. So in your great big collection of CD4 positive T cells, they're all directed against different peptide fragments. Now, of course, some of them will end up being directed against the same peptide fragments, but the idea is that you have a huge variety of different CD4 positive T cells that are directed against different peptide fragments. But you will have some in your massive great collection that happen to be directed against the same peptide fragment. And we would refer to those CD4 positive T cells that are all directed against the same peptide fragment as a certain clone of CD4 positive T cells. So to now use the proper terminology, in your body you will have a huge number of different CD4 positive T cell clones that are all directed against different peptide fragments, and hopefully none of which are directed against peptide fragments that actually appear in your own proteins. That's the idea anyway, but it does go wrong. So, in an individual who is going to develop inflammatory bowel disease, so let's just draw this little individual. So here is a little stick person, who is going to develop inflammatory bowel disease. And a little bit of information for you. Inflammatory bowel disease generally comes on in the second or the third decade of life. So you don't usually get it when you're 5, for instance. Usually it comes on either in the teenage years or in the 20s. So that's the normal period when people actually first develop inflammatory bowel disease. So in this individual who's going to develop inflammatory bowel disease, what has to happen if we're going to have a failure of tolerance, we're going to get the adaptive immune response attacking a commensal within the intestine, is that you have to, for some reason, develop CD4 positive T cells that are directed against peptide fragments of one of the commensals within the intestine. Okay, so I'll say that again. If this individual is going to develop inflammatory bowel disease, for some reason, he is going to end up getting CD4 positive T cells that are directed against a peptide fragment of one of the commensals within the intestine. And depending on which commensal this is, will determine whether he gets Crohn's disease or ulcerative colitis or one of the rarer forms of inflammatory bowel disease. But in short, it will determine which type of inflammatory bowel disease he gets. So the question then is, why does he end up developing an immune system that can attack the commensals within the intestine, and other people do not develop an immune system that can attack the commensals within the intestine? And the answer is, I can't tell you the answer to that. We don't understand that at present. What we do know is that genetics plays a part. So your genes are going to determine which CD4 positive T cells you end up with. So I'll put it feeding into this. So it determines which clones of CD4 positive T cells you end up with. In addition, it's not just genetics. Because if it was genetics, then identical twins, if one identical twin developed inflammatory bowel disease, the other would also have to develop inflammatory bowel disease, and we know that that's not the case. You can have identical twins where one of them has inflammatory bowel disease and the other doesn't have inflammatory bowel disease, so it's not purely genetics. So there must also be environmental factors which determine which clones of CD4 positive T cells that you actually develop. And probably a degree of it is just luck, how your immune system develops, whether it all works perfectly and you don't end up with CD4 positive T cells that are directed against peptide fragments of the commensal microorganisms. Now, in most people, there must be mechanisms for making sure that you don't end up with CD4 positive T cells that are directed against peptide fragments of commensals within the intestine. We don't fully understand what those mechanisms are, but for genetic reasons and environmental reasons, in someone who is going to develop inflammatory bowel disease, those mechanisms fail and you do end up with CD4 positive T cells that are directed against peptide fragments of one of the commensals within the intestine. And just coming back to this genetics here, we do know that genetics plays a part, because if you do have two identical twins and one of them has inflammatory bowel disease, there is a massive great chance that the other will develop inflammatory bowel disease as well. It's not 100% though, which is how we know it's not purely genetic. However, that chance is significantly higher than if you have non-identical twins. So if you have two non-identical twins and one of them develops inflammatory bowel disease, the other has a much lower chance of developing inflammatory bowel disease than they would if they were identical twins. So genetics does play a role, and we think it's many genes that will influence which clones of CD4 positive T cells you actually end up producing. So, your genetics and environmental factors determine your immune system, and by your immune system I mean which clones of CD4 positive T cells you're going to actually end up having within your body. And in most people, well, in fact, in all people, there are mechanisms to prevent you getting CD4 positive T cells that are directed against peptide fragments that are present within the proteins of the commensals within the intestine. However, this slips up in inflammatory bowel disease. In an individual who's going to develop inflammatory bowel disease, they are going to end up producing CD4 positive T cell clones directed against some peptide fragment of one of the proteins of a commensal microorganism, and this is how they're going to end up developing inflammatory bowel disease. Okay, so we will have a break here. In the next video, we will see how having this clone of CD4 positive T cells that is directed against a peptide fragment of a protein of a commensal microorganism within the intestine is actually going to lead to inflammation occurring within the intestine, i.e. inflammatory bowel disease.